In the cold, deep waters of the Arctic Ocean lurks a mysterious, slow-moving beast. They've lived in the shadows for hundreds of millions of years. But an incredible new discovery has shone a scientific spotlight on the Greenland shark. Julius Nielsen, a marine biologist from the University of Copenhagen, and a team of fellow scientists found some of these creatures could be as old as 400 years. By far, the longest living vertebrates ever found. Alive before America's independence, the reign of Napoleon Bonaparte, and Beethoven's First Symphony. The discovery has surprised scientists from around the world who study animals and aging.
It is pretty amazing. We don't expect a vertebrate to live nearly that long, and that's the point of why we're so shocked. We just thought that vertebrate physiology would prevent that kind of possibility.
Scientists can typically determine an animal's age by rings in their bones, but because shark skeletons are made out of cartilage, Nielsen and his team had to find an alternative. They chose the eyes. The lens of the eye is made up of proteins that build up over time, similar to the layers of an onion. The proteins at the very center of the eye develop when the shark is in utero. Over 3 years, researchers collected samples from 28 Greenland sharks that had been accidentally captured and critically injured during an annual fishing survey. Once the sharks were euthanized, their eyes were removed and sent to a lab where scientists separated the center for testing. By carbon dating that layer of the eye, they were able to estimate the shark's age.
It took accumulating samples of this very unusual unknown organism over a long period of time to get that data, which is revolutionizing our idea about how long vertebrates can live, how long tissue can be healthy.
The data reveals the sharks could live much longer than other vertebrates, almost double the previous record holder, the bowhead whale, and trailing behind, the Aldabra giant tortoise, the beluga sturgeon, and humans. Raising the question, what is it about the shark's biology or environment that could help us understand why we grow old when we do? So far, little is still known about why the species lives so long. It could be because they live in icy waters, or because of their sluggish metabolism. But scientists have been studying other animals for answers. Dr. Sarah Cohen has spent her career studying tunicates, or sea squirts.
Okay, so here are our closest invertebrate relatives. These lovely gelatinous blobs here are sea squirts. They're bags of water. They also feature a stomach and a filtering basket.
Tunicate cells can regenerate so quickly they appear to bypass aspects of the aging process entirely. In one experiment, a researcher found some species could regrow from a small clump of cells in as little as 6 days. They might look nothing like us, but their proximity to humans in the evolutionary tree means their molecular processes are similar to ours. Dr. Cohen believes that makes them valuable research subjects for understanding human aging.
They're gene networks, and the molecules that control aging processes are much more similar to our own human processes and those of other vertebrates, in contrast to another colonial animal like a coral, which is potentially immortal, but extremely different from us in terms of the level of complexity of tissues.
Dr. Cohen's research shows that tunicates in the wild regenerate differently from those in the lab, proving that although regeneration is hardwired, it is also affected by external factors. Their similarity to humans indicates the same could be true for us.
So this incredible institute is the Buck Institute for Research on Aging.
Dr. 𝗥𝗲𝗹𝘁𝗲𝗀 is an expert on the biology of aging and is trying to understand how and why humans age.
The more complex the organism is, the harder it's going to be to tease that out. But we have the tools to do it. This is not beyond the realm of possibility. Is it a good experiment? All right, well, let's take a look and see.
She studied cell senescence, a process where a cell decides to stop dividing. It typically protects the body from cancer or promotes healing, but as we age, these non-dividing cells build up.
They become what has been termed maladaptive. They're doing the bad thing. They're causing a problem called inflammation, which is a sort of low-level immune response that underlies virtually all the major diseases of aging.
Diseases like Alzheimer's, arthritis, and cancer. While normal cells look like this, senescent cells are larger and irregular in shape.
Here's one large, flat, senescent cell. Here is the nucleus. Normally this cell would be maybe a half or a third of this area.
Studies in mice show that as senescent cells begin to accumulate, diseases we associate with aging, like heart and kidney failure, increase. But eliminating these cells isn't the obvious answer. A team of researchers from Mayo Clinic made senescent cells disappear in mice. The mice were healthier for longer, but there was little change in their maximum lifespan, suggesting other factors are also driving aging.
And it's a big question for us. What causes aging? What causes the death of tissue? And we're realizing from the sharks and from other organisms that different things are acting in different ways in different organisms.
Evolution is telling us something about complexity and what it would take to extend the lifespan of a complicated organism like a person. And I think we're very close to that evolutionary limit, and we really need to understand evolution better.
Well, the secret to extending life is extremely complex. Scientists are now unlocking important clues that could allow us to lead longer and healthier lives. And animals such as tunicates, mice, and now the Greenland shark will continue to play an important role in that research.